day, viewers. This is 300 Plus Academy, where all we do is exams tutoring, ensuring that you ace your next and every exam. Still on the business of biology practical for 2024 Neco examination. We've looked at group 1, talking about specimens A, B, C, onion bulb, ginger rhizome, and Irish potato. The link to that lecture video is in the description. We've looked at specimens D and E, talking about bean seedling and corn seedling as it relates to epigeal germination and hypogea germination. The link to that particular lecture is also in the description of this video. Right about now, we're going to be looking at specimen F, G, H, I, and J. That's talking about group 3. When we looked at this, we have a link between specimen I and J, and then there is a link between specimen G and specimen H. And then we have pigeon's head with neck just somewhat standing alone. So we'll look at how we can put the puzzles together. So what should our question exactly look like? Let's take off with specimen I and J. As you can see in the question on your screen, the question says, list 3 features that adapt specimens I and J to their environment. List 3 features that adapt specimens I and J to their environment. Specimen I here is a cactus plant. And when we talk about cactus, you know, cactus is a xerophyte, in that it is basically those are plants that, you know, they live where there is little or no water, let's say like the desert, okay? And the problem here is always about how to conserve water and how to get water so that they continue the process of photosynthesis. And specimen J here is water lettuce. This is an hydrophyte. Where the problem with specimen I, cactus plant, is about water conservation. How do they conserve water? That is the problem with the cactus plant. In the case of the water lettuce, the problem is always how do they quickly get rid of excess water that has been absorbed by the adventitious root. So it's just like opposite problem. One is facing the problem of drought, and the other one is facing the problem of excessive absorption of water, or you say excessive water intake. So in that question, it states that we should list 3 features that adapt specimen I and J to the environment. When we talk about specimen I, the first one is possession of spiny leaves. Now, when we say spiny leaves here, you know, the more the surface that the leaf presents to the sun, the more it is exposed to water loss, or what we call transpiration. So in a bid to cut down on transpiration, in a bid to cut down on water loss, cactus, as you can see in the diagram on your screen, the leaves has been reduced to spiny leaves, thereby helping in water conservation. Also, the stem is very thick succulent stem. Thick in the sense that when water is absorbed, the water stays inside and it is juicy in there, so that no matter the heat from the sun, the water cannot actually be lost from the plant via transpiration, via the lenticels and the stomata. Also, possession of thick succulent leaves. The leaves of cactus plant, talking about specimen I, is very thick, and not just that, it is also cuticularized. That's, it has a waxy layer all over it, so that it cuts down on transpiration greatly, thereby conserving water are needed for physiological processes in the plant. So these are about the three features that adapt specimen I to its environment. Also, you can talk about the root system. The root here is, that kind of root that is very deep. Possession of fibrous roots so that it enables them to absorb water even in the desert and the hot region where we can find specimen I. Talking about specimen J, we find them mostly covering the surface of water bodies. So what are their adaptation to survive on top of water where they float? Possession of broad thin leaves. We see broad thin leaves. That's just opposite of this. Where this is trying to conserve water, this is trying to lose water. So when the leaf is thin, it now means water can be lost from the leaves of that plant by transpiration very easily. And that is what we want here, so that the leaves of the plant is not saturated with water. So possession of broad thin leaves, possession of air spaces for buoyancy. Yes, the plant in this case needs to float so that it is not submerged in water. When it floats, it can still obtain carbon dioxide from the surrounding atmosphere and photosynthesize. So we talk of buoyancy here, possession of air spaces within the plant for buoyancy. Also, numerous adventitious roots, so that even when they float on water, the adventitious roots can help to absorb water, most especially mineral salt absorption. So these are just about the three features that adapt specimen J, talking about water lettuce to its environment. So that just answers that. Question 2 states, identify specimen G and H. We were just told to identify the specimen G is actually the atlas vertebra. Specimen G is the atlas vertebra, and specimen H is the lumbar vertebra, as you can see on your screen. Specifically, it is that of a rabbit, but the important thing is specimen G is atlas vertebra and specimen H is lumbar vertebra. When we say atlas, atlas is the very first cervical vertebra just immediately after the skull. Axis is the second cervical vertebra. The atlas and the axis form the atlantoaxial joints needed for nodding movement. Then we have the lumbar vertebra exactly at the back region. So for this question, it says identify specimen G and H. We've been able to do that. State one distinguishing characteristic. So when I said G is atlas vertebra, what is the distinguishing feature or characteristic that I saw? When you talk about atlas, the thing is it has no vertebral body. You can see that diagram, it has no vertebral body. Normally, a vertebra bone is supposed to have a body, but in this case, there is no body. It does not have a vertebral body. Also, it has no spinous process. Yes, it has no spinous process because around the neck are not very much muscle is really attached to the cervical vertebra. So talking about specimen G, it has no spinous process. It is very clear in the diagram on your screen. Look at what a vertebra with spine, with spinous process looks like, and look at what the atlas vertebra looks like. The spinous process isn't really conspicuous, it's not really there. So it has no spinous process. So seeing these two, it was very easy for us to identify that that was atlas vertebra. Now, talking about the lumbar vertebra, the distinguishing feature is that the vertebral body is large. In fact, of all of the vertebral bone, it is the lumbar vertebra that has the largest body. The vertebra that the body is large and very prominent. Also, the spinous process is broad and massive for attachment of the muscles of the back. So those are the distinguishing feature for lumbar vertebra. So moving on to 2B, it says state 4 differences between specimen G and specimen H. As you can see, the differences right there on the screen. When you talk about the atlas, which is a type of cervical vertebra, it has a reduced transverse process. It's in the neck region. So, and the transverse process is not really there because the muscles there that needs to attach to that bone aren't all that much. So reduced transverse process, but for the lumbar vertebra, you can see the long and large transverse process for attachment of the muscles of the back, like trapezius and the likes. Also, for the atlas or the cervical vertebra as it is, I have said it here, no spinal process, no neural spine, or you say small spinal process or small neural spine. But for the lumbar vertebra, the neural spine is broad and massive, and that's exactly what you also have there. Also, for the atlas, it has a very wide neural canal so that the spinal cord descends from the head region into the spinal bone, into the vertebral column. So the canal here is wide, but as we move down from the cervical vertebra to the sacral vertebra, you see that the neural canal narrows down. So the neural canal for the atlas vertebra is actually wide, but for the lumbar vertebra, it is narrow. Also, we have vertebrae canal present for passage of blood vessels around the neck and head region. You look at it, vertebrae canal present. You can see that also in the diagram talking about the vertebrae canal. But you can see the name vertebra, then arterial, vertebrae canal present. In lumbar vertebra, there is no vertebrae canal. For the atlas vertebra, a centrum is absent, but for the lumbar vertebra, centrum is present and well developed. You can as well see that in the diagram. Then, since the atlas and the cervical vertebra generally, they are not really attached to much muscles, we say facets, attachment facets are few. The facets for attachment to muscles are few, but in the lumbar vertebra, the facets for attachment to muscles are more, and that just about ends that. So moving on to the next question, we have question 2. So they said, in what part of the body is each of specimen G and H found? So where can we find the atlas vertebra? Where can we find the lumbar vertebra? Very simple. The atlas vertebra can be found on the neck, while the lumbar vertebra can be found in the back region. Also, we have another question. What type of skeleton is specimens G and H? Now, talking about specimens G and H, they form a part of the axial skeleton. Recall that when you talk about the axial skeleton, it comprises of the skull, the vertebral column, and then the rib cage, which comprises of the sternum and the ribs. And also you have the appendicular skeleton, but most specifically, the atlas and the lumbar vertebra, talking about specimen G and H, they are part of the axial skeleton. Look at the question. It says, what type of skeleton is specimen G and H? They are axial skeleton. When you talk about the appendicular skeleton, you're looking at the upper limbs and the lower limbs. So we have another question here. It says specimens G and H are found in between what bones? What this question means is that what bones come before specimen G and what bones come after specimen G. Then what bones comes before specimen H and what bones come after specimen H. Talking about the atlas vertebra, the bone that comes before the atlas vertebra is the skull, and after the skull, that's when you now have the atlas. So the bone that comes after the atlas now is the axis. Look at the question again. Specimen G is found in between what bones? Yes, it is for the atlas vertebra. It is between the skull and the bone immediately after the atlas vertebra is the axis cervical vertebra. So the skull and then the axis. Now, when you talk about the lumbar vertebra, the bone that comes before the lumbar vertebra is actually the thoracic vertebra, and the one that comes after the lumbar vertebra, talking about specimen H, is the sacral vertebra. Next question says, make a label drawing of the anterior view of specimen G. Anterior view of specimen G. You recall a biology practical exam is not complete without drawings. In the case of group 2, we made drawing about a potted bean seedling. For specimen for group A, if I am told to speculate what you might likely be drawing, that's a difficult one. But I think I'll be going for maybe specimen B, ginger rhizome, maybe or maybe not. But we've attempted this here. Now, looking at this one, we were told in these questions to draw the anterior view and label as well. Make a label drawing of the anterior view of specimen G, 8 to 12 centimeter. The sort of diagram you actually need to draw is actually what you can see displaying on your screen. Look out for the various parts, the neural spine, the neural canal, the vertebraterial canal, the facet for occipital condyle, the odontoid fossa, so that the dens of the axis vertebral is just going to attach there. Yes, those are just about the drawings there. Let's look at it once again. So what should the drawing look like? That is exactly what you can see on the screen, but for time constraints I would not be sketching. But what your diagram should look like is exactly what you have on the screen. You can see the numbers, the number labeling for you to see the various parts you need to label. So it's important to get your pencil, pause this video once you get to this diagram, and try to sketch the atlas vertebral. So what does one represent? One here represent the neural spine. Two in the diagram represent, that's the central neural canal for the passage of the spinal cord. Then you have vertebraterial canal for passage of blood vessels. You have the odontoid fossa, that's talking about, for when you say the odontoid fossa, there is what we call the dens of the axis. So the dens of the axis is actually meant to enter into the odontoid fossa so that it aids the nodding movement of the head. Then you have the anterior tubercle, talking about label 5. Then for 6, you have the anterior arc. Recall that this is anterior view of specimen G. Then you have the facet for occipital condyle, where the skull actually attaches to the atlas vertebra. That is talking about 7, facet for occipital condyle. And then 8, you are looking at the wing shape cervical with wing-shaped cervical rib. And then 9, you have the posterior arc. Look at the diagram very well. That is exactly what you should try to sketch for the atlas vertebral. Looking at the next question, we were told to give one function each of any 3 parts that we have labeled. We should give one function each of at least 3 parts that we have labeled. Talking about the parts are labeled, if you actually get to work and make sure you draw that diagram talking about the atlas vertebra and you label Now, what you're being told to do for question 2G is to state functions of at least three parts label. When you talk about the neural canal, it is for the passage of spinal cord. When you talk about the vertebral canal, it is for the passage of blood vessels. When you talk about the odontoid fossa, it is for articulation with the dens or odontoid process. You can see that on your screen. Immediately after the atlas vertebral is the axis, and the axis has what we call the dens or odontoid process that inserts into the odontoid fossa of the atlas vertebra. So that is what is going to aid nodding movement. Also, talking about facet for occipital condyle, it is for articulation with the skull. That's this particular facet talking about occipital condyle is where the skull articulates with the atlas vertebra, and that answers that. So the next question states give three functions each of specimens G and H. So what are the functions of the atlas vertebra? What are the functions of the lumbar vertebra? So talking about specimen G, the atlas vertebra, it allows us to nod and rotate our head. It allows for nodding and rotational movement of the head. It supports the skull because the skull directly rests on the atlas vertebra. It protects the spinal cord and vertebral arteries. Another function is that as the spinal cord descends from the medulla into the neck and then to the back, the atlas vertebra protects the spinal cord. Also, the vertebral arteries around the neck region is being protected. You can see the vertebral canal. It protects the arteries as well. Also, it provides points of attachment to different muscles of the neck. That is what G does. It provides points of attachment for various muscles in the neck. Talking about functions of specimen H, recall that H is lumbar vertebral and the location is actually at the back. So it bears the great weight of the body as they occupy the center of gravity. Most of the weight of the body actually rests on the back. Then also the lumbar vertebra maintain proper gait and posture of the body. Also, the lumbar vertebra, since it bears the most of the weight of the body, it supports pregnancy in females, and that just about ends the functions of G and H. If you have more, you can add, you can remove some, you can add the ones you have, and you'll be good to go. About specimen F, a pigeon's head with the neck intact. So, what are the probable questions we should be looking at for specimen F? Look at the very first question we have. That's question 3. It says, name the organism to which specimen F belongs. Name the organism to which specimen F belongs. So the answer there is going to be a pigeon. And so A Roman figure 2, it says, state the phylum, class, order of the organism mentioned. So since it's a pigeon that this specimen F belongs, now the phylum is actually Vertebrata, which means animals with backbone. The class is aves, and then the order here is columbiforms. So that answers that particular question, columbiforms. That answers that particular question. For A Roman figure 3, 3A Roman figure 3, it says states 3 characteristics of the class to which F belongs. Now, F belongs to the class A. So we're told to state 3 characteristics. The first one, I said the body is covered with feathers. The mouth is modified into honey toothless beak or bill. You look at the mouth in the diagram on your screen. The mouth has been modified into a honey toothless beak or bill. And also, you need to understand that aves are warm-blooded, which means they are homoeotonic. They are warm-blooded organisms. So that ends that particular question, 3A. Moving on to 3B, we were told to state the habitat of specimen F. Now, the habitat here, talking about the pigeon, is actually terrestrial, or you say we can find them in parks. We can find them on the top of city buildings. So those are the habitats or places where we can find specimen F. So moving on to the next question, that talks about 3C. In question C, we were told to state how the structure of specimens F adapt the organism to their mode of life. Recall that they are terrestrial. You can find them at the top of high-rise buildings, city buildings. You can find them at parks. So we are told to look at the structure of this specimen F, those structures that adapt it to its mode of life. Now, when you look at this question, we're told to look out for the structures. So this is more like saying what are the observable features or structures that adapt specimen F to its environment. Now, when you look at the image on your screen, talking about specimen F, it has two nostrils or nares on the beak for breathing. Exactly on the beaks, there is this tiny hole. It is called nares, or you call it nostrils for respiration, as for breathing. Two, the toothless beak is for obtaining food, for scratching, through the down feathers around the neck, for cleaning, for repairing the feathers, and it also for collecting nesting materials. When they want to build their nest, they use the beak to collect nesting materials, and it is used as weapons against enemies. Talking about specimen F, it has eyes. So that eye is for what? Keen vision, and the eye has the ability to see colors, allowing them to watch out for predators for easy escape. Also, when you look at specimen F, and Fs generally, they are very flexible around the neck region. They can turn their head up until like 180 degree. And the reason for this is because in humans, we have about 7 cervical vertebrae, and that the highest turning we can is not even up to 180. But for birds, on the average, they have about 13 cervical vertebra in the neck region, so that they can look directly behind them, so that even if they have predators coming right behind them, they can turn their neck and they can see. So the neck is very flexible. It's another way or adaptation for survival according to its mode of life. And that answers question 3C. Look at it very well. Flexible neck, a bird can turn its head 180 degrees so that it can look directly behind, so paving way for easy escape from its predator. And the reason is very simple. It has more cervical vertebra, an average of about 13 in the neck region. So that ends that question, talking about observable structures that adapt specimen F to its mode of life. Still on specimen F, can we be told to draw this? Biology practical is not complete without drawing, although I do not think so. But just for the sake of completion, the drawing is showing on the screen. You can see the eye, you can see the frontal head, you can see the nostril, you can see the beak, you can see the throat, you can see the hair covert, you can see the in neck, and that just around that region. So just in case you're told to draw specimen F and label, that's the labeling for you on the screen. Most importantly, the head, the nostril, the beak, the throat, the external ears, the neck, and then the head. That's just about the important area. So you want to try and practice those drawing. Okay, that ends that. Okay, for the next question, E. We were told to state the mode of feeding of F. State the mode of feeding of F. So when you talk about F is actually heterotrophic, more specifically omnivores. Omnivorous. So it is an omnivore. So in the question, you can see we were told to state the mode of feeding of specimen F. So it is actually heterotrophic, more specifically that it carries out omnivorous nutrition, more like an omnivore. So sometimes it's going to feed on nuts, plants. Sometimes it's going to feed on insects. So that's why it's more specifically an omnivore. So that answers that. So moving on to the next question, question F, we were told to name one type of food that the organism that possess F consume. The organism that possess F is a pigeon. So what type of food does it consume? It consumes grains, it consumes legumes, seeds, nuts, worms, and insects. No wonder it is an omnivore. That's omnivorous feeding. It is heterotrophic nutritionist, and that ends that. Okay, moving on to question G. Question G says that state the type of feathers on the neck of specimen F. What type of feathers do we have on specimen F? Well, on specimen F, the type of feather that we have, that's talking about question G, is actually down feathers. Down feathers is specifically useful in maintaining body temperature. Remember that the class apes are homeothermic animals. They are warm-blooded. So the kind of feathers on specimen F is actually the down feathers, and it is there for maintaining body temperature, for keeping the organism, the specimen here, warm. And with that, we have come to the end of the third group, talking about the specimen. Thus far, about 10 specimen has been treated. So the link to the previous group, talking about group 1, group 2, is going to be in the description of this lecture video. And do stay tuned for the concluding group, talking about specimen K to O. From myself and the entire team, it's bye for now.